Hello everyone, my name is Panlu, and today I'm going to demonstrate to you our EPIC Immune Atlas platform. EPIC stands for Extended Phenotypic Immunome Characterization. So in this platform, we have a database which consists of 114 healthy individuals, ranging from cord blood samples to newborn individual samples and all the way to adult samples. So in this platform, scientists or researchers from all over the world are able to upload their which is their mass cytometry data, onto our platform and run through our pipeline analysis automatically and get to compare it against our database. So before any comparison is to be made, let us look at our database first before we start to explore the analysis process. Users could click here to view the healthy database. And here shows our database of 140 samples. So this shows the volcano plot. And we can double click to zoom into full screen. So after we zoom in, we can have a clearer understanding of the volcano plot. So for example, a certain node, say node 35 and age 12. And then we can tell the cell frequency at this point is about 4%. And also the cord blood, there's this very high intensity being observed at the cord blood, about 15% at node 15. So users could look into our database very carefully and look at the cell frequencies in each age group. So let's get back to our platform. Apart from the volcano plot, here shows that relative abundance plot. These nodes are grouped according to their node frequencies, so users could observe what are the patterns and the phenotypes as well. So phenotypes will be shown here, as you could see from the screen. These are being automatically generated from our pipeline. Users could also compare this result with the previous heat map shown in the previous page. These are the SOM codes and they are grouped according to their cell types. So each color now represents the cell types. And here shows the t-SNE plot of our 114 samples, and each color represents the cell type. Users could look into our database in this way as well. There are some reclustering being done because according to the SOM t-SNE plot and PCA plot as shown, there are some nodes which are overclustered. So regroup them, do the reclustering, and show in different colors as shown here. Okay, here shows also the relative abundance, but instead of the nodes, it shows the cell types. And also, after that, 3D volcano plot again will be shown on our database. Instead of nodes, here shows the cell types. So you could observe what are the cell types which are high in which group. So these are our database results. So let us zoom in to take a closer look at the volcano plot. So the same as before, we can look very closely at the volcano plot after we zoom in. So as cell type is plotted against the age group, I want to look at what type of cells are being expressed highly in each age group. Say, for example, if I look at adults, I could see that, there's a 27% of memory CD4 T cells in adults, which is higher than any age group. So for the cord blood as well as the naive T cells. So we can look at individual ones very carefully. Let's get back to the main panel and look at other plots. Here shows the cell proportion changes across the age group. So from the left, cord blood newborn, all the way to adult. And each color shows specific cell type. So you could see the cell type proportion changes across different age groups. And lastly, here shows the proportions of each cell types across age groups in a more separated way. For example, the B cells starts to increase when the age increases. So this gives a clear background and knowledge about our database. So now imagine I am a scientist X, and now I'm going to upload my data and compare with the current database. So I'll do some uploading of the data, which I have already done, and there'll be some loading process. The analysis pipeline on our platform will be carried out for my data, and it will be compared against the current database. So here shows the result. So I'm using HPV samples. As you could see from here, these are the phenotype differences in terms of the cell count. And also here will show me the distance plot between my samples. And here are the database results. When my samples are added to the database, and the marker's distance will change as well. And the importance of each marker will change according to this plot, as well as the NRS plot. And also the t-SNE plot of the samples will be plotted, and each color shows each age group, and here shows my result and here shows the database results. And also the media expression of the markers, and a recommended K according to our choosing criteria. For my case, it will be 87 nodes. So I will look into here and then see whether is there any overclustering. Say for here, there's some overclustering of the nodes because different colors, which represents different nodes, are clustered together, even though they are separated as different nodes. So I'll look into their phenotypes separately using this heat map. So to aid our t-SNE dimension, we also look at the PCA dimension. And after I look into the marker expressions, I will upload a data sheet like this. So there will be two columns. One shows the node ID, the other shows the cell phenotype. And it will automatically generate for me the results here. It will show me how my HPV samples in each node are compared against the other age groups in our database, and also the relative abundance in each group when my samples are combined with the healthy database. The colors will be reassigned according to cell types in the SOM t-SNE dimensions as well as the PCA dimensions, such that I will look at them separately. When you move your mouse over, you will also show the cell types that you upload in your datasheet previously. So the color are determined by the datasheet you uploaded. The t-SNE plot of the cell types will also be shown. So these are my HPV samples, and this is the database. As I only uploaded 2 patients with HPV, so I will select their age group. They are all of age 14, so I will look at age 14. So I will look at how the cell types changes across different cell types, as well as just in age 14 samples. So for instance, I look at the memory cells and the CD8 memory cells. And also, the pipeline will do automated reclustering after some merging according to the cell types you have identified. And also, you will show the relative abundance again, but in terms of the cell types, and the lineage marker expressions as well as the functional marker expressions. One good thing about the functional marker expression is that, let's say when I click on the adults, it will automatically arrange the marker expressions according to their values. And these are the markers with low expressions, and these are the markers with high expressions. So this will tell you further information about their marker details in specific their expressions. After this, I'll look into the 3D volcano plot, and it will tell me what the cell types being observed in my age group. So for instance, I could see that in my age 14 HBV samples, the double negative T cells are very high, and also the B cells. And I can do a direct comparisons with the database here. And also, the cell proportions as well will be shown here, and I will look into my own age group over here, which is the HPV samples here. So I can compare directly, not just in terms of the age group, so I'll compare against my age 14 samples over here. I could also compare across all the other healthy samples against my HPV samples. And lastly, this shows a similar information as compared to above. And if I want to look at my HPV samples, I could see very clearly that each cell type proportion difference in my samples as compared to the healthy samples in age 14 as well as across all other age groups. You could see that my naive CD8 T cells is much lesser than those ones over here, and also the other cell types. This is the end of the presentation, and thank you for listening. Hope that you have now a better idea on what our analysis platform can do for your project. Feel free to contact us if you have any questions. Goodbye.